In the last lecture, I motivated neuroscience as a discipline in part by pointing out that animals and humans have many features in common, from molecules all the way up to the entire brain. And the similarity between us and with animals, both in terms of our cellular structures and also our behaviors, make it possible to study brain function in non-human animals. And I want to come back to this theme. It's a recurring theme in the study of neuroscience. So there are universal similarities among brains, and we can see these by comparing the structure, for instance, of a rat brain and a human brain. And a rat brain and a human brain share anatomical architectures. These architectures are shared across species, especially across related species. Now, as it turns out, a rat brain and a human brain, in fact, are fairly similar when one looks at the entire family tree of life, more similar, say, for instance, than a human versus a squid. If we look at the entire architecture of a rat brain and a human brain, we can see these universal similarities. These brains look fairly similar either to an untrained eye and also to a neuroscientist, and I want to show you some of these components here. Now, we can see here that these two brains are arranged in approximately the same layout. We have the cerebellum in back, we have the brainstem in the center. And then we have the neocortex, this large part of the brain that is also shared among us and a rat's brain. If we look at these two brains, we can see that these structures are arranged in similar relationship to one another, similar position. They're connected to one another. And if you look very closely, those connections are also very similar. And so if we look here, we can see, for instance, that the hypothalamus sits below the principal part of the brain, both in a rat and in a human. We can see that in the brainstem, in the brain's core, we have the pons and medulla. These are structures that are important for core functions in both humans and in rats. And if we keep on looking, we can see these. And in each case, we can see similar structures in human and in rat. And the thing that's different is perhaps the size or perhaps the relative proportion of these things. These gross sizes and relative proportions are the ways in which humans and rats differ. In other words, in many respects, it's a question of differences of degree as opposed to some qualitative difference in the basic brain plan. So we can see just by looking at these anatomical diagrams that mammalian brains share a lot in common. And so that's one kind of universal similarity that we can see between humans and rats and other mammals. Now, another kind of similarity is at the cellular level. And I pointed out before in the previous lecture neurons, one that I filled with dye. Nervous systems throughout the animal kingdom are made of neurons, and in many cases there are also glial cells. And these neurons and glia are the fundamental building blocks of brains across the animal kingdom. And at the level of single cells, these neurons and glia have analogous physiological functions. And what that means is that when you record from a neuron, whether it be from a squid or from a rat or in some cases from a human, you can see similar physiological events. These similar physiological events are the universal currency of brain signaling. So, for example, you can take a neuron and stick an electrode into it. And it is possible to stick an electrode into a neuron and measure electrical activity. And it's possible to do this in a variety of animals. Easier to do in a squid because the neurons are larger, but it's possible to do in a wide variety of animals. And what you see when you record from a neuron is you put the electrode in, and you see that the voltage is a little bit negative compared with the outside. It typically rests at about minus 70 millivolts, so that's a small fraction of the voltage, say, on a 9-volt battery. It's less than 1% of the voltage on a 9-volt battery. And this resting potential is where neurons begin. And then when you inject a little bit of electrical current into the neuron, it makes a little bit of a voltage change because that's the way neurons operate. And if you inject enough current, what happens is that suddenly you get this large event, this spike-like event in which the voltage goes up and comes back down and does so in just a thousandth of a second, a millisecond. And that event is called the action potential. It's also called colloquially a spike. And these spikes are the fundamental currency of how neurons work. And these spikes occur in us, in squid, and it turns out that the fundamental principles by which spikes operate, the way that they work, is shared between us and squid. And what that means is it's another aspect of the universality of brain architecture and brain layout and functional principles, that even down to the level of single neurons, there are these shared principles that are universal across the animal kingdom. Now, as I said, when comparing the rat and the human, there are differences among brains, of course, and one of the most salient differences is the number of neurons in a nervous system. So, for example, a small nervous system like one can find in a small worm might only have a few hundred neurons, and so that would be a very small nervous system. One might find a larger nervous system in an insect. Insects typically have thousands or hundreds of thousands of neurons in the case of a big insect like a grasshopper. And then when we get to vertebrate animals, we start getting larger nervous systems. So for instance, in a mouse, one might expect to find hundreds of millions of neurons. Looking in us, we're getting into tens and hundreds of billions of neurons, hundreds of billions of neurons in 100 billion neurons in humans, and hundreds of billions of neurons in a whale. And so one principal difference among animals is not the types of cellular components, which are the same, but the numbers of these components. Okay? Now, just as neurons are similar in many ways when you look across different animals, it turns out the events of synaptic transmission are also preserved. The ways that neurons talk to one another are preserved when we look across all these animals. This principle of synaptic transmission is something that's found across all animals. And if you look even more closely, you can find molecules that are very similar in their protein structure, again, between us and a squid. In fact, if you were to take ion channels, which we'll be hearing about from a squid, and we measure the properties of those ion channels in squid have similar properties, and when you heat up those ion channels, they start behaving a lot like mammalian ion channels. And so what that means is that life is similar at the molecular level. This illustrates a principle called the principle of common descent, the idea that these signaling molecules are the same because they had a similar ancestor, and that similar ancestor is reflected in the diversity of animal life. Now, those are similarities that are seen across the animal kingdom. There are, of course, also dissimilarities. One dissimilarity is that proteins are, in fact, not identical. And so the proteins that we find in, say, the membrane of a squid neuron and ours are not really exactly the same. Another major dissimilarity is that genes are regulated. So the ways that genes are regulated, perhaps a gene gets turned on and you make a certain amount of protein. That amount of protein that's made or the place it gets sent is a little bit different. And so there are regulatory steps that are not identical between animals. And finally, as I indicated, bigger brains have more neurons. Bigger brains are also more complicated, and these larger brains have perhaps more parts because they're larger. Their proportions may be different. For instance, in us, we have quite a lot of neocortex compared with a rat. Our brains are something like 80% neocortex, whereas a rat is only about 40% neocortex. And so those are examples of differences that we can see among animals. Despite these differences, there are general principles that have emerged from animal work that are applicable to the human brain. Now, let's look at some of these general principles in terms of two commonly held popular beliefs about the brain. Many people have these particular popular beliefs about the brain, and I'll list them. The first belief is that your brain is like a computer, commonly held belief. Another belief is that you only use 10% of your brain. Now, what I'd like to do is examine the function of neural tissue and think about whether these two beliefs stand up under the scrutiny of what we know about brains. The first one, your brain is like a computer. Well, the brain is not like a computer. Now, as it turns out that the brain is very well laid out in many ways, we will go over how the brain is very efficient energetically and only uses 15 watts of power. And that's an amazingly small amount of power, comparable to an idling laptop. But the brain is not like a computer. It's not that efficient in certain ways. It's efficient energetically, but what the brain really is a survival machine. It's optimized over millions of generations of natural selection and evolution to help you survive. It helps you live to fight another day. It's not there performing some kind of supercomputing operation and leading to some, calculation of pi to 100 digits or what have you. What the brain is doing is helping you survive to fight another day. It's helping you to reproduce, to breed, to live and fight. And so in that respect, the brain does not need to get things accurate. It does not need to report events in a precise fashion. What it needs to do, rather than be a truthful reporter of events, is it needs to help you survive. And so one theme that we're going to be returning to is the idea that your brain, in fact, tells you lies. Your brain lies to you. Your brain misrepresents, distorts, all in the interest of making sure that you can survive. And that's a central principle of how brains are organized, and we're going to be talking about that later in the course. Now, the second belief, and this one is one that is worth examining in terms of both neural function and also popular belief. And the question is, how much of your brain do you really need? This is what I would call the 10% myth. This is perhaps the most persistent popular belief about the brain. In surveys, this belief is prevalent around the world. You can see it in bus stop ads in the United States. You can see it in Brazil. There's a survey that shows that something like half of Brazilians believe that they use only 10% of their brains. And so this is a belief that has worldwide reach. It not only is popularly believed, it has cultural consequences. And so if you look in movies, you can see entire movies built upon this motif. And these movies don't even need to explain the idea. So for instance, there's a popular movie, Defending Your Life, in which the person comes back to Earth and has to live again and again and again until he is able to use a little bit more than 10% of his brain. There's another popular movie, The Wedding Crashers, that makes fun of this belief, where the guy says, I believe we only use 10% of our hearts. And in that case, the belief doesn't even need to be explained. Everybody knows that 10% is how much of our brain we used. It's helpful in thinking about this kind of thing. It's used as a motivator to help us use our brains a little bit better. So for instance, a common version of this belief is Einstein used 15% of his brain. If only we could use 15% of our brains, we'd be like Einstein, that kind of thing. And so this 10% belief permeates the culture. It's used to motivate us to do a little bit better. It's used to help us reach our full potential. It's even used to explain paranormal phenomena, the idea that perhaps psychic abilities come from this other 90% of our brain. Now, it's worth thinking about exactly where this belief came from. If we look back through historical writings, we can perhaps see evidence of this. So if we go back and look through the history of neuroscience and psychology, one place where we can find this is in the writings of the pioneering psychologist and pre-neuroscientist William James. William James gave public lectures in which he said to audiences, we only reach a small fraction of our brain's full potential. And that, I think, is inarguably true, that we do not reach our full potential. So he made that statement to public audiences as he toured the United States, and that was what he said, and that was circa 1908. Now, it turns out this claim then mutated. The first recorded instance of this claim about 10% of the brain came from someone who is not a scientist, Dale Carnegie, the well-known self-help guru. And what Dale Carnegie said was that we only use 10% of our brains. He used this to win book sales and influence readers. And this appeared in the introduction to that book. It was an introduction written by a man named Robert Lovell. And Carnegie liked to quote William James as being the source of this quote. He liked to say, well, James said it, he's a scientist, and therefore it must be true. And so that's the earliest recorded mention of the idea that we only use 10% of our brains. Now, that's the belief. I've dwelt on it, but let's think about the actual facts. I've mentioned to you different ways of understanding brain function. One is looking at lesion evidence to see what goes wrong when some part of the brain is damaged. Another thing I've mentioned is ways in which we can monitor brain activity. When you put an electrode into a brain, or when you scan a brain, say in an fMRI or a PET scanner, you can see what parts of the brain are active. And these kinds of evidence, lesion evidence and imaging evidence, have thoroughly demonstrated that every part of the brain is necessary for normal function. And this, as it turns out, goes back in time. And if we look back historically, we can see evidence for this particular framework for understanding brain function going all the way back to the Roman physician Galen. Galen, who lived during antiquity, did animal experiments, and also it turned out that he had a day job as a physician treating gladiators. And he found that, in fact, gladiators who suffered some particular injury to some part of the brain lost some particular function. And Galen was able to observe that every part of the brain was necessary from, as I said, treating animals and also gladiators. Now, a classic example of localized function comes from perhaps the most famous neurological patient in history, a railway worker named Phineas Gage. Now, Phineas Gage was a railway worker who had a nervous habit. His job was to tamp holes with a tamping rod and plant charges and then blow up those charges. So his job was to tamp down charges. He had a nervous habit. His nervous habit was that he took his tamping rod and he sharpened it just against objects. And he made this nice sharp tamping rod that was blunt at one end and then sharp at the other end. And one day he was doing his job, and the tamping rod was in the hole. And it blew through his prefrontal cortex. It went right through here, came out up here, and it basically made a hole in his prefrontal cortex, right? So after that injury, he survived, amazingly, because the tamping rod was sharpened to a point, because it went right through his skull. So he survived, which was very fortunate for him. But what was curious was that he changed. He was not the same guy. It turns out that before the injury, he was a hardworking man. He was a good friend to his colleagues. He was a really solid guy. After the injury, he became an irresponsible layabout. He was unable to keep a job. He was unable to keep friends. He became itinerant. He basically lost what we would call self-control, what we now call executive function. It turns out that it's possible to associate these losses of function with particular brain injuries because his skull survives, and it's possible to do computer reconstructions to see what exactly was wrong with his brain. This tamping rod went through his skull and destroyed frontal regions of his brain. And these frontal regions apparently are responsible for the qualities of character and of diligence that he had, but then lost after his injury. And so it appears that these prefrontal parts of the brain are necessary for executive function. Now, what's interesting as a historical footnote is that recently a portrait of him has been found. After his injury, he went on tour across the United States with his tamping rod, and you can see him in this portrait very proudly posed. And you can tell that it's him for several reasons. One is that the eye is closed on the side of the injury. Another is that there's a carving on the tamping rod, and this carving matches the carving that's on the tamping rod in a museum, and so it's possible to match this tamping rod. I should say that this portrait originally was interpreted as being perhaps a whaler holding his harpoon, but it turns out that, in fact, it's Phineas Gage. And so we now are able to look at this man and see what he looked like and see what his character appeared to be like even after his injury. He was a very strong and willful man, and you can see this in his face. Now, Phineas Gage is perhaps the most famous example of a neurological patient, but it's possible to see other kinds of deficits throughout history in people who have had brain injury in other parts of their brain. And it's possible to localize different brain regions to being necessary for various kinds of functions based on lesion data, based on brain injury data. And just to name a few examples, for instance, when spinal cord is damaged, we lose sensation or perhaps the ability to move our limbs. When we have different parts of our neocortex damaged, we lose ability to do various things. The neocortex is composed of different parts, roughly speaking, frontal, parietal, temporal, and occipital. And when each of these brain regions is damaged, we can see specific losses, functional losses, that are associated with damage to that particular part of the brain. I mentioned the frontal part of the brain in Phineas Gage. For instance, in the parietal lobe, we start neglecting parts of the world, or perhaps we lose our ability to do manipulations like math and arithmetic. In the occipital lobe, when the occipital lobe is damaged, then we start losing the ability to see things. Occipital lobe damaged patients become blind in various ways. And that's an example of brain injury being associated with a specific loss. Another brain region is the cerebellum. The cerebellum's back here. And the cerebellum is important for coordinated movement. It's important for processing sensory events, to generate movements, to generate smooth movements, or perhaps to learn a tennis stroke, that kind of thing. The cerebellum is an important structure for sensory processing to guide movement. And in patients with cerebellar damage, those patients lose the ability to do these things. And finally, the hippocampus. The hippocampus is a structure that appears to be important for navigating in spatial environments, for learning new spatial environments, and also for factual recall in humans. And there's a famous patient named HM. HM was a patient who had not damage to his hippocampus, but he had actually brain surgery done to treat intractable seizures. And after that surgery, which removed parts of his brain that included his hippocampus and also structures that were near the hippocampus, HM lost the ability to form new memories. These are all examples of localization of brain function that we now understand based on examining patients who either had brain injury or perhaps had some surgery done to treat some condition. So those are examples of brain localization. So in summary, you need every part of your brain. You require 100% of your brain. Now, the fact that you need your entire brain and the fact that your entire brain is called into service during the course of a day means that it is of interest to monitor what parts of the brain are active at any particular moment. And today, it's possible to monitor brain activity in a living, intact person with whole brain imaging methods. And the two most popular methods are functional magnetic resonance imaging, also known as fMRI, and positron emission tomography, also known as PET scanning. And these particular forms of brain scanning are useful as research tools, and they also tell us that every part of the brain is called into service at one time or another. Now, how does fMRI work? As it turns out, brains use a lot of the body's total energy budget. Even though the brain is a very energetically efficient thing, it also is an energy hog relative to the rest of the body. The whole body uses about 70 watts of power. The brain uses about 15 watts of that power. And what fMRI reports is it reports a reflection of that consumption of energy. And specifically what fMRI reports is increases in blood oxygenation. So what happens is, if we look at this image of an fMRI signal, what we can see here is that fMRI is reporting increases in blood oxygen. What we see here is a pseudocolor in which the purple and yellow and red regions indicate regions of increased blood flow. And these increased regions of blood flow reflect increases in blood oxygen, and that's a consequence of neural signaling. Now, if we stop and look at this for a moment, there's something that's not quite right, which is that I'm telling you that fMRI monitors neural activity, right? But what we're looking at here is increases in this blood oxygenation. This is a signal that reflects blood oxygen. So why would it be that the fMRI signal shows increases in blood oxygen despite the fact that neural tissue is more active? Shouldn't it be that neural activity, in fact, leads to decreases in blood oxygen? So there's a paradox here. And that paradox is that it's not the neural activity and it's not the oxygen consumption itself that leads to the signal. So what's really going on here is that the way the fMRI works is that neural activity triggers blood flow. Now, it's not exactly known how, but there is an event in which synaptic activity, communication between neurons, is thought to trigger events that lead to increases in blood flow. And so what's going on here in this signal is that there's blood flow that comes as a result of neural activity. And so the idea here is that synaptic activity, communication between neurons, leads to a signal that gets sent out to the blood vessel. And then the blood vessels then show increases in blood flow. And that increase in excess of what is needed at the moment leads to a signal that we can see in fMRI. And so as a result, the blood flow goes up. In some sense, it's in anticipation. So that's the way the fMRI technique works. Now, if we think about this, we can think about this in terms of energy efficiency. Now, I said that brains are very efficient despite using a large share of the body's energy budget. It turns out that the amount of energy used by the brain is similar to, as I said, an idling laptop or perhaps the light inside your refrigerator. In some sense, we are all dim bulbs. Now, this amount of energy consumption is constant. It turns out to be constant in waking life. If you're thinking about something really difficult, you still use 15 watts. If you're asleep, you use still 15 watts. The only circumstance in which the amount of energy consumed by the brain is reduced is when you're in a deep coma. In deep coma, the energy use of the brain is something like more like 7 or 8 watts. It's reduced by about 50%. And so as a result, the brain is always using the same amount of energy, and there is some sense in which it appears to be running flat out all the time just to maintain its capacity for generating electrical signals. And so one way to think about fMRI perhaps is that fMRI is a call for energy where it's needed. So if the entire brain is using 15 watts, perhaps fMRI reflects synaptic activity saying, well, I need energy over here, and so you should send some blood in my direction. And so perhaps what the fMRI signal is telling us is it's showing us where blood flow is going, perhaps in anticipation as a need for future activity. All right? And so that's really what the fMRI signal is. It's in some sense a call for energy for future tasks and perhaps for immediate future needs. Now, fMRI is a useful tool for giving a general sense of what brain region is active. It has a resolution, as you can see in the image, of about 1 millimeter, perhaps a few millimeters. And so it gives us a general sense of what part of the brain is active. Now, fMRI studies are done in a very carefully constructed manner where you have someone do a task and then you compare that with a very similar task and you ask what's different between the tasks. So for instance, a typical fMRI study might be one in which you have someone read words, and then the comparison would be the person is just looking at a screen and doing something that involves all the stages of reading except for reading itself. And that difference would be then the thing that a neuroscientist would study. And that difference allows us to see which brain regions are recruited when a person reads. And so therefore, fMRI studies tell us what's different when we do some particular thing, such as reading. And that's a very interesting finding. That sort of thing is very useful in understanding where perhaps brain regions that are specialized for reading are localized. So that's a thing that fMRI can tell us. It's very interesting. It's not necessarily the whole story. So it's not the whole story for several reasons. One is that I just told you that it measures differences between reading and some other tasks. So in fact, what it shows us is what's different. It doesn't show us all the other things that brains are doing while the reading is taking place. Another thing that fMRI doesn't show us is its resolution is only about 1 millimeter, and its resolution over time is only a few seconds. And so fMRI does not give us details of processing. It doesn't give us exact circuit mechanisms for what a particular brain region does. It doesn't give us details of processing on a moment-to-moment basis. And so there are things that are outside fMRI's current resolution. So fMRI is a powerful tool, but it doesn't really tell us everything that we want to know about, say, how a circuit might compute something. Now, one feeling that you might get from looking at these scans is that some particular brain region is activated during a particular task. And in some sense, that might tell you that, well, okay, that region is lit up, so in fact, that region is what is responsible for reading. And I would call that a modern version of the 10% myth, because as I mentioned before, you need your entire brain to do things. At any given moment, you're using large components of your brain. If we knew everything that was happening in the brain during reading or whatever task we might be trying to understand, we would find that there was activity throughout the brain. And so fMRI gives us a partial snapshot of brain function, not really the whole picture. It gives us an important way in to understanding brain function, but it's by no means the whole story. Now, let's summarize what we've learned in this lecture. So what I've told you is that across species, brains have similar components, from synapses up to gross brain structures, and these similar structures are present across the entire animal kingdom, from flies and worms up to humans. Mental functions in all animals require many parts of the brain working together, not 10%. The brain uses a lot of the body's energy, but it's still a very energetically efficient device, and this energy efficiency allows the possibility of imaging brain function. It allows the possibility that energy availability and the provision of energy can be used as a tool for imaging brain function, and that blood flow is triggered by a call for energy triggered by synaptic activity, and that call for energy, the response to the call for energy, can be monitored in the form of scanning techniques like fMRI.